Okay. So as I mentioned that our lab works in prostate cancer. Today, I'm going to talk about a new finding in the lab. It's totally an unpublished data. We have communicated this manuscript, but it's a very clinically relevant data that we have. It's an observation that started in the clinics, and then we worked on the basic science part of it. So the title of my talk is Supraphysiological Testosterone and Engaging the Immune System in Prostate Cancer. Next slide. I don't have any disclosures. Next slide. So the objective is to understand how supraphysiological testosterone causes growth inhibition in prostate cancer. So this was the first manuscript, and it was a seminal manuscript, an observation by Charles Hopkins way back in 1941. He published this finding in the first volume of Cancer Research. The title of his finding was how androgens drive prostate cancer. So this was the first observation that suggested that androgens can drive prostate cancer growth. And by stopping androgens, you may be able to stop prostate cancer growth. For that, Hopkins got the Nobel Prize. So if you can go one slide back, Lalita. This work, one slide more. So this work was then continued by his mentee, Susant Kachhap, who is better known for the active metabolite of testosterone, which is Ph.D. And he had noted, and this was also noted by Charles Hopkins, that androgen, although it causes prostate cancer growth, and inhibiting androgens either by surgical castration or by chemical castration. So they used to use estrogen way back then. The growth of prostate cancer can be inhibited. However, during that time, one thing was noted by Hopkins and by Susant Kachhap was that the androgens also causes a paradoxical growth inhibition of prostate cancer. So can I have one more slide? This was noted by Susant Kachhap. He had done lots of work on this, but it was also reproduced elsewhere across various labs. And it was not only reported as a lab-based phenomena, but but also in the clinic. So this is a clinical trial done in 1967, where high doses of testosterone is given to a patient with prostate cancer. And back then they didn't have PSA, so they are measuring acid phosphatase. And what you find is that when you give high doses of testosterone to the patient, the acid phosphatase level drops down substantially. So what that means is that you would have a decrease in the prostate cancer growth. And this is exactly how we measure biochemical recurrence by measuring PSA. So next slide, please. So here at Hopkins, Sam Demid and colleagues, what they did was they used this same strategy. However, they, instead of using one dose of the high doses of testosterone, what they did was they had repeated cycles of testosterone, and they call this therapy as bipolar androgen therapy. The reason why they call it as bipolar androgen therapy is the patient sees testosterone at a very high dose, and then the testosterone level is allowed to decay, if you will, to the near castrate level. And then again, you would have another dose of testosterone. So there is this repeated cycle of high dose and low dose of testosterone that the patient sees. And this particular therapy has been remarkably good for a subset of patients. If you can go to the next slide, please. It is a well-tolerated therapy, so it preserves the quality of life. Next slide. And as I mentioned, that in a subset of patients, you have this remarkable regression of prostate cancer, and some of the patients remain disease-free for years. Now, if you go one slide back, please, thanks. So here is a patient who has responded dramatically to this therapy, and what you find is when you give testosterone to this patient, there's a drop in PSA, and the patient is disease-free for months. Now, this is the time when Sam came to our lab and we got involved with this particular clinical study. What we noted is that in the tumors of these patients, you have an increased propensity for DNA repair gene mutations. So our simple hypothesis for that was, if you can go one slide more. There's androgen causes DNA double-strand break, and this was known. From work here at Hopkins, as well as from work by Arul Chinayan's group at Michigan. And if you give high doses of testosterone or synthetic or the active metabolite, or even synthetic testosterone to prostate cancer cells, what you find is that there is this DNA double-strand breaks. So this is measured here in this slide by using gamma H2AX staining, which is a surrogate marker for DNA double-strand break. And what you find is that the H2AX foci increases when you have high levels of androgens. Now, no one really knows exactly how that happens, but probably it is transcriptionally induced with topoisomerase-induced DNA breaks, because as the androgen binds to the androgen receptor and transcribes its gene, what happens is there is this constraint on the DNA which has to be released, and topoisomerases might be able to do those stress relief for those particular areas or particular genes, and then you have this DNA double-strand breaks. So next slide, please. So our simple hypothesis way back then was that supraphysiological testosterone induced this DNA double-strand break, and cells which harbor DNA repair gene mutations might be susceptible because they may die of DNA repair. They would be unable to repair their DNA damage and might die of DNA repair crisis. So next slide, please. So surely enough, in the lab we tested these 3 different prostate cancer lines, which were androgen receptor positive, and which had varying degrees of DNA repair gene mutations. So LNCAP cells have the most. So it has mutations in the BRCA2, ATM, MSH2. These are all DNA repair genes, and they are DNA repair genes for various DNA repair pathways. So VCAP cells have intermediate DNA repair gene mutation, and LAPC4 cells have one DNA repair gene mutation, but that which is reported in the cosmic database, we didn't find it here at Hopkins when another lab here at Hopkins had sequenced for those particular mutations. So if you look at how they respond, these 3 cell lines respond to supraphysiologic androgens, you find that the LNCAP cells respond to it with growth inhibition. So it's the blue line in the graph in C. So that's how the LNCAP would respond. And then if you look at LAPC4 cells, which has the least DNA repair gene mutations, they actually like androgens and they grow better. The next slide, please. So we wanted to know whether this is true, and we repeated those experiments, and we were able to reproduce those results. So this is a clonogenic assay. It's a long-term survival assay, and what you're looking at are 2 different prostate lines, and the LNCAP cells are sensitive to androgen, and indeed they were sensitive, and the LAPC4 cells are insensitive to androgen, and they like and are not growth inhibited. So naturally, the first thing that occurred to us was maybe these cells are dying with apoptosis. And we looked at PARP cleavage, which is a marker for apoptosis, and we didn't find any substantial increase in PARP. There's some increase, but not to account for that growth inhibition. So the next pathway that we looked for is necrosis. And what we found was we looked at several necrotic markers and we didn't find any change in necrosis. So probably the dying of a cell death, which is non-apoptotic, non-necrotic. So we looked at other pathways. So next slide, please. One thing we knew about these two cell types is that both of them respond to androgens by inducing autophagy. The LN-cap cells are different than Lap-C4 cells. The LN-cap cells have a greater induction of autophagy, and the basal level of autophagy in Lap-C4 cells doesn't change. We didn't find a whole lot of other autophagy genes going up, so probably the whole transcriptome of autophagy is not upregulated by androgen, but selective autophagy happens in these, not selective, but the existing autophagy machinery is used by these cells in response to androgen. Now, just having an increase in autophagy by looking at this LC3B signaling doesn't really mean autophagy has increased. In order for autophagy to happen, the whole process of autophagy has to take place. So as you may be aware, autophagy is a process wherein you have autophagosome forming, and they have to be degraded into by fusing to the lysosome. So this whole process of autophagy has to occur for us to say that autophagy has increased. So we looked at this autophagy flux by using this sensor, which is LC3 molecule, which is labeled with both the red and the green signal. So when you transfect this sensor in the cells, what you end up is having LC3 puncta as both red and green. However, when the autophagosomes fuse to the lysosome, the acidic environment quenches the green signal, and you end up with only the red punctus. So if you look at the LN-cap cells, what we had found was that there's an increase in the double positive puncta, but there is also an increase in autolysosome. It's the hybrid organelle that when autophagosomes fuse to the lysosome, and that doesn't happen with the Lap-C4 cells. So what this told us is that the whole process of autophagy actually increases in the LN-cap cells in response to androgen. So next slide, please. So then we looked at whether there is any connection between autophagy and some non-apoptotic cell death, and indeed there was a connection, and the cell death mechanism was called as ferroptosis. As the name suggests, ferroptosis is a cell death mechanism. It's a non-apoptotic cell death mechanism which depends on iron. So iron is highly regulated in the cells. So iron is available through transferrin, and as it comes inside the cells, it is then taken up by ferritin, and it is stored there and is made available to the cells wherever iron is required. However, ferritin can be degraded by a selective process called as ferritinophagy, and that's a selective autophagy process. And the way in which it happens is ferritin is can I go back? Ferritin is degraded by autophagosome, but it needs an adapter called as NCOA4. So as NCOA4 binds to ferritin, it ferries it to the autophagosome. One slide. So the first thing we looked at is what happens to ferritin. And surely enough, in the sensitive cell lines, we found that ferritin gets degraded with increasing concentration of testosterone. Can I go? Okay. So what happens to the adapter molecule? The adapter molecule is induced in the sensitive cell line, and it doesn't occur in the Lap-C4 cells, which is the insensitive cell line. The other thing that we noted was whether the interaction between the NCOA4 and ferritin occurs in response to androgen. And we did this with the immunoprecipitation experiment, which is shown in the plot below. When you add testosterone, what you find is that the NCOA4 is induced, but it also binds to the ferritin, and that's the blot on the lower blot on the right hand side. So NCOA4 does bind to ferritin in a SupraT responsive manner. What happens to this ferritin is the next thing we investigated. So we looked at autophagosomes in these cells, and we looked at ferritin. So what we find is in response to SupraT, ferritin localizes with the LC3B puncta, which is the autophagosome. So it happens only in the sensitive cell line and doesn't happen in the non-sensitive cell. Can I go one slide? Right. So at the end of, so the mechanism by which ferroptosis occurs is this ion, which is released when ferritin is degraded. That ion causes damage to lipids by forming this lipid peroxides. And the buildup of lipid peroxides then causes cell death. So if you look at the control cells, they do not have so much of lipid peroxide, and the way in which we measure this is by using C11 BODP, which is a lipid peroxide sensor. And under the influence of SupraT, you do see an increase in lipid peroxides. So ferroptosis is a very highly regulated process. So there are genes that regulate ferroptosis. There are pro-ferroptotic and anti-ferroptotic genes. So we looked at a bunch of those genes, and we found that all of them are increased in the sensitive cell line, and that doesn't occur in the non-sensitive cells. In order to prove that this is, go one slide, Max. Thanks. In order to prove that this occurs, the cell death mechanism is through ferroptosis. We used a ferroptotic inhibitor, and here in this case we use ferrostatin 1. And in the absence of the inhibitor, you do see that there is an increase in cell death when you give testosterone in the sensitive cell line, and that's the blue bars. But when you combine it with the ferrostatin 1, what you find is that there is a decrease in the cell death, so that the cell death is abrogated. So next slide, please. So the summary so far is that SupraT induces ferroptosis. It induces ferroptosis because it can degrade, it uses autophagy to selectively degrade ferritin, a process called as ferritinophagy. So next slide. So this was the question that we asked next. And the question was whether the autophagy occurs because there is DNA repair stress, because these cells are unable to undergo DNA repair, as a result of which you would have stress building up, and probably that triggers autophagy. So we tried to look at that by dual staining the cells for autophagosomes as well as for the DNA double strand break. So here the double strand break is gamma H2AX, the red puncta that you see, and the green one is for autophagosomes. We found exactly the opposite to what we had hypothesized. So cells which are repairing which are unable to repair the DNA after damage with SupraT, those cells have less of autophagosomes, and the cells which were able to repair their DNA during this repair process, they had more of autophagosomes. So there was an inverse correlation between the number of autophagosomes a cell harbors and the DNA damage. So that posed a new question to us, was whether autophagy is utilized by these cells to actually clear the damaged DNA. So it can be either it is helping in the DNA repair process, or autophagy being a degradative process, whether this damaged DNA is shuttled outside in the autophagosome for degradation is the next thing we asked. So the first thing we looked at is whether such a thing exists in the natural world. So next slide, please. So surely enough, there is something called as nucleophagy, and it was first discovered in yeast. And in the yeast, the lysosome, which is the vacuole for yeast, that degrades a part of the nucleus. So it pinches a part of the nucleus and the contents are degraded there. So whether something like this occurs in the mammalian system, yes, it does occur in the mammalian system. So it is also present in humans. So humans who have mutations in the nuclear lamins have a nuclear envelope which is not, which the integrity is compromised, as a result of which the nuclear material leaks out, and that can be degraded by the autophagosome, which is the green vesicle that you see next to the nucleus. So we then asked whether that is happening in these cells. And to do that, what we did was we had to use autophagy inhibitor to stop this process of autophagosomes fusing to the lysosome, because if the fraction of autophagosomes that carry this DNA is less, we won't be able to see it just by staining it with DAPI. So we stop that process of fusion, and we allow those autophagosomes to build up in the cells and look under the microscope to look at whether we see signals of the nuclear material in the cytoplasm. And surely enough, in the LN cap cells, we do see the cytoplasmic DNA, which was less in the V cap cells, which is the intermediate cell line, and we didn't see any in the Lapsifer cells. So one slide back, okay. So the next question was, what is the origin of this DNA? So it could be a mitochondrial origin, although mitochondria doesn't stain with DAPI, but it might be that that in the autophagosomes, if you have this mitochondria building up, because autophagosomes also degrade mitochondria, you may end up with signals in those autophagosomes. So we looked at mitochondria by staining for a mitochondrial protein, and we looked at the DAPI signal together. We didn't find any strong co-localization of the DAPI with the mitochondria. So it told us that it is not of mitochondrial origin. However, when we stain the same cells with autophagosomes, we do find a very high co-localization of the DAPI signal with the LC3B signal, and that's in the lower panel. So we do have cytoplasmic DNA, and that cytoplasmic DNA is present in the autophagosomes. In order to prove whether that is the damaged DNA, we actually stained the cells with H2AX and looked at whether the cytoplasmic DNA that we find in the autophagosome, whether that is also H2AX positive. And surely enough, it was H2AX positive, which meant that it is the damaged DNA that is shuttled out for degradation. Next. So we also looked at whether the DNA is present in the lumen of the autophagosomes. And the way in which we did was we looked at the intensity of the LC3B signal. And as you can see, that the red dots, when they go down, that's where the lumen of the autophagosome is, and that's exactly where the DAPI signal peaks. So it is present in the lumen of the autophagosomes. Next slide, please. So in summary, this part of the talk, we did find that supraphysiological testosterone increases ferritinophagy. It increases ferroptosis, and it actually unleashes two autophagy-mediated processes. One is ferritinophagy, the other one is nucleophagy, where damaged DNA is shuttled to the cytoplasm for degradation. This was exciting for us because what it told us is that the cytoplasmic DNA might induce nucleic acid sensors present in the cytoplasm. Next slide. So that might also induce innate immune signaling. So two of the labs which have done very seminal work on these nucleic acid sensors are Glenn Barber's and James Chen's lab. Together, between the two, they have identified the nucleic acid sensor in the cytoplasm, which is the C gas molecule. And they also identified the adapter that is needed for relaying the information to the innate immune genes. So just to give you the pathway. So next slide, please. So this is a cartoon, so you may have to click it. So DNA in the cytoplasm is recognized by cGAS, which is an enzyme that creates this cGAMP molecule, which is a nucleotide. This cyclic nucleotide is then detected by STING, which is present on the surface of the endoplasmic reticulum. As it binds to cGAMP, it dimerizes, moves out into autophagosomes like vesicles, where it acts as an adapter to bring in TBK1, which is the central kinase, which is the central innate immune kinase. And then what it does is it phosphorylates downstream molecules, which are transcription factors. And as they are phosphorylated, they would move into the nucleus to carry out innate immune gene transcription. Now, two of the molecules that are central here, transcription factors, are IRF37 and NF-kappa B pathway. Next slide, please. So we looked at the adapter molecule, which is central to the signaling, and STING increased with increasing with SupraT over time in the LN cap cells, which is the sensitive cell line. It doesn't happen so with the lab C4 cells. The other thing that I told you is that STING would move from the endoplasmic reticulum to the autophagosomes like vesicles, and the blots on in panel B there is a sucrose gradient where we break down the cells and we separate the autophagosomes by using the gradient. And now we are looking at STING and the autophagosome. And you can see that with the presence of testosterone, SupraT, what you find is that STING gets induced, but it is also co-fractionates with the autophagosomes. Now, I told you that STING, when it becomes active, it dimerizes. So the gold standard for STING activation is this dimerization assay, and it's a native gel where you look at the dimerization of STING, and what we find is that in the presence of SupraT in the sensitive cell line, you do get STING induced, but it also dimerizes, and that doesn't happen with the lab C4 cells. One thing that came as a surprise to us back then, and now it's no more a surprise, but was it doesn't only induce the DNA sensor, but it also induces the RNA sensors in the cytoplasm. So cells have sensors to detect both the DNA and the RNA, because you could have RNA or the DNA virus coming in. So that is seen as a threat. So the RNA sensors are the RIGI and MDA5, and we did find that RIGI and MDA5 are induced by SupraT. They are not only induced, but they also are activated, just like how we had the STING getting dimerized. We have this adapter molecule, MAVS, that gets oligomerized on the mitochondria after activation. So with SupraT, you do get oligomerization of MAVS, and that doesn't happen in this insensitive cell line. Next slide, please. So we looked at what would be the mechanism. We initially thought that maybe androgen receptor, being a transcription factor, might induce some of the retroviruses, which are the endogenous retroviruses, and that might be detected by this RNA sensors. We looked at the motifs for androgen receptors in these latent endogenous retroviruses, and we did find some motifs in some of the retroviral genes. However, when we looked at the transcript level of these genes, we didn't find much change in any of these genes, which told us that this may not be the mechanism. So we went back to the literature to find whether activation of STING, whether it would also activate the RIGI pathway. And surely enough, there was publications, and we can go next slide. So James Chen's lab and other labs have found that activation of STING by the DNA in the cytoplasm can also activate the REGI pathway. And the exact mechanism is not known, but it could be by the transcription, RNA pol 3 based transcription. You would have these RNA being transcribed, and that might activate the REGI pathway. So what we had found was that there is nucleophagy, and that would activate the nucleic acid sensors. Downstream of that is the central kinase for innate immune signaling, and we tried to look at whether that is activated. Surely enough, that was activated, and this is an activation phosphorylation mark for TBQ1, and it was activated in the Ellen cap cells. We didn't find the transcription factor which TBQ1 phosphorylates, which is the RF3, and that's the chief transcription factor that that would get activated. And we didn't find any of those phosphorylation. So we looked at the dimerization for RF3 because it also dimerizes when it gets activated, and we didn't find any RF3 dimerization. However, we did find an activation of the other transcription factor, which is RF7 in the Ellen cap cells and not in the Lapsifer cells. So the next thing was TBQ1 also activates the NF-kappa B pathway, which is another central innate immune signaling pathway. We did find the canonical subunit of NF-kappa B, which is the P65 unit, getting phosphorylated, and that activates, that gets activated in the Ellen cap cells. Now, activation of these molecules would lead to their accumulation in the nucleus. So the blot in below in I, panel I, is a nuclear extract, and you can see that between as you treat the cells with supraphysiological testosterone, you do have any accumulation of IRF7 and NF-kappa B in the nucleus. So just having them in the nucleus doesn't mean anything. Whether they are activated is the next thing we looked at, and this is a luciferase assay for NF-kappa B activity. And under SupraT conditions, you have an activation of the NF-kappa B pathway in the sensitive cell line. It doesn't occur in the non-sensitive cell. Next slide, please. So this was the canonical NF-kappa B pathway. It's the most studied pathway. However, the understudied pathway is the non-canonical NF-kappa B pathway. And we looked at the central kinase for this particular pathway, which is the NIK. And that phosphorylates the P100 molecule, which is another subunit of the NF-kappa B pathway, but the non-canonical subunit. So once that gets phosphorylated, it undergoes degradation by the proteasome. The processing doesn't degrade the whole molecule, but it processes the P100 molecule to a P52 molecule that moves along with RLB inside the nucleus to activate the non-canonical pathway. And we did find activation of the NIK and the processing of the P100 molecule, as well as an induction of the RLB molecule. The other thing that we noted was that whether the processing has any meaning, whether these actually move inside the nucleus, and here we did it with the immunofluorescence, and we did find that the P52 molecule increases in the nucleus. So what this told us was that the canonical as well as the non-canonical pathways, NF-kappa B pathway, are activated by SupraT. So downstream of this are all the innate immune genes, and we looked at some of these innate immune genes initially. So all of them were upregulated in the sensitive cell line, and they were not upregulated in the non-sensitive cell line. One of the molecules that was highly upregulated was CXCL10. So this is a chemokine which has receptors on NK cells as well as T cells. And that was highly upregulated at the transcript level. So it was not only upregulated at the transcript level, but next slide, please. But it was also upregulated at the protein level. So this is protein inside the cell, which is the lower middle blot. But it is also secreted in the media, which is the top blot, under supraphysiological testosterone conditions. And that doesn't occur in the Lapsifer cells. In order to prove that these immune genes are indeed activated by the STING regi pathway. We knocked out several of these subunits. So we made CRISPR-Cas knockouts for STING, which is the DNA sensing pathway. The regi, we made double knockout of STING and regi. We made knockouts of the central kinase TBQ1 as well as the essential subunits of the canonical and non-canonical pathway. Using these knockouts, we looked at CXCL10, and we did find that CXCL10 goes down with the double knockouts strongly, and with the knockouts for both the canonical and non-canonical pathway. One thing that we noted was that if you knock out canonical pathway, the expression level of CXCL10 just gets abrogated. So probably that must be the essential pathway. On the slide, please. The next thing we did was we looked at whether the canonical pathway is the essential pathway and whether that drives the non-canonical NF-kappa B pathway. So we looked at the processing of P100 molecule to P52 in the knockouts. So in the wild type, it occurs in response to testosterone. That is totally abrogated in the P65 knockout, or the knockout of the central immune kinase, which is the TBK1 kinase. Also, the stabilization of NIK, which is the central kinase for the non-canonical pathway, that gets abrogated when you knock out P65 or TBK1 or double knockouts of STING and regi. In order to understand the immune genes which are regulated by this pathway, we used an anostring analysis. So we looked at 770 immune gene signatures. We came up with 52 of those which are induced in the P65 STING or the canonical, either the canonical or the non-canonical pathway. So in a STING TBK1 NF-kappa B manner, these genes are induced, and on the left is the nanostring data. On the right is we looked at a bunch of these genes and validated it with real-time qPCRs. So in summary, this part of the talk, what I have is that SupraT induces autophagy, and using the autophagy, it induces two different autophagy mediated pathway, which is ferritinophagy and nucleophagy, and that is able to induce the innate immune signaling through the nucleic acid sensors. Next slide, please. So what about the immune cells? So this first we did some in vitro analysis. So this is an NK cell migrating towards the tumors, towards the prostate cancer cell, which are treated with testosterone. And this is an in vitro assay where you have cells in two different chambers, and you're looking for migration of those cells, immune cells, to the chamber where those tumor cells are. And there was an increase in migration of the NK cells towards the tumor cells. We also did some in vivo analysis by looking at tumor xenografts of the sensitive cell line. And in the control, you find that there's not so much of autophagy induction. We looked at using, again, the LC3B signaling. What you also find is that there's an increased expression of CXCL10. That's the chemokine that would attract the NK cells. And surprisingly, in the panel B, we did find cytoplasmic DNA in the autophagosomes in these tumors. This is without any treatment with chloroquine. So we didn't have to really stop the autophagosomes fusing to the lysosome in this case. So the dynamics might be different in vivo. The next thing we looked at is whether it can attract any of the NK cells. So these are nude mice. They don't have the T cells, but they do have a very robust NK cell activity. And so if you look at the control tumors, they have few NK cells infiltrating. However, when you treat it with testosterone, you do have a very high infiltration of CD57 positive NK cells. And we looked at it not only with one marker, but we also used other markers. One more. So we also looked at it with a different marker for NK cells, which is CD49B. And you can see that those are the control tumors, and these are the treated, and there's an increased infiltration of NK cells. One slide, please. So those are the animal data. What about the clinical data? So we had from Sam Denmead and colleague, Sam Denmead's clinical trial, as well as there are other colleagues who are conducting this clinical trial, Emmanuel Antonakaris, and then Mark Markowski. So they had biopsies from patients who received this therapy. So this is pre-treatment. You can see that the tumor is very evident in those biopsies. And with post 3 cycles of that, you do see that the tumor is propagated or almost gone. So we looked at these tumors for the immune cell infiltration. One thing I wanted to tell you is prostate cancer is an immune desert. That is one of the reasons why immunotherapy doesn't work so great in prostate cancer. So the control biopsy is on the right-hand side, and the same person is treated with testosterone. And you can see that there is increased infiltration of cytotoxic T cells. So that's where we are right now. We are also looking at different immune subsets in these tumors by using a multiplex analysis. This is in collaboration with Angela DiMarzo here at Hopkins. And we are excited to share that data. We have some initial data which looks very exciting. So in summary, what we have is supraphysiological testosterone might induce ferritinophagy as well as nucleophagy, and might induce these nucleic acid sensors, both the DNA and the RNA sensors. And downstream of that are these immune genes, which might lead to an increased immune infiltration in these tumors. And that might be one of the mechanisms by which these tumors are growth inhibited. And that might specifically occur in the cells which have DNA repair gene mutations, because nucleophagy might be more evident in those cells. So at the end, I want to acknowledge my lab for doing this. It was most of the work was done by the 2 postdocs, Janet Mendonca and Rajendra Kumar. Now, but I'm also blessed with a number of undergrads who work tirelessly to raise all those knockouts, because it's a very laborious process to raise all those knockouts. So they were very instrumental in doing those. Sam Denmead is my clinical collaborator. And then I also have collaborations with Drew Padol, who did some of the immune related studies, and then Angela DiMarzo, who helped us with all the pathology studies. And these are my findings. And thank you for your patience. And sorry about the delay.
Thank you, Susant. That was just terrific. You finished up in really good time, leaving us with a few minutes for discussions. I'd like to open this for discussions.
Hey, Susant, can I ask you a question? So your cell line model is interesting. You used initially LinCap, VCap, and LACP4, and you mentioned about BRCA mutation and ETM and all that. But is there any way you wanted to check or want to check now that there is this X gene rearrangement in those 2 cells, and your treatment massively induces these transcription factors? Is there any role for those things potentially? Because we know that this X transcription factors indeed induces H2AX staining and all that. We didn't follow it up, but what are your thought on that?
So you're talking about the ETS rearrangement, right?
Right.
Okay. So that work was initially done here at Hopkins, as well as done by a group in Michigan, that these DNA double-strand breaks by androgen might induce those rearrangements and gene fusions. So this was done, I think, in 2008. And that was one of the things that put us into this DNA repair pathway. So right now, at least in the lab, I'm not doing that, but I know that in the clinical samples, we didn't have any correlation with the H2AX, with the ETS fusion. So, but we did find a very high correlation with the DNA repair gene mutations.
Yeah, I had a follow-up question in the chat box. When you say about these treatments, are the patient previously sequenced to confirm the BRCA and ATM mutation and then the treatment started, or?
No, so these are all retrospective things. We've had these archived samples, and but that's a very good question because we are thinking of stratifying the patients who would get more benefit with the treatment.
Yeah, that's a good question, Suri, because I was thinking of that as well, about stratification. Dr. Shu Feng has a couple of questions.
Okay, I have two questions. First question is the big picture question. So, very interesting finding. I think, do you have any evidence or do you suspect that what you find, the mechanism you find with, this estrogen can also applicable to other sex hormones such as estrogen breast cancer?
That's a good question. So, so way back in 2008, when this was reported that androgens induce double-strand break, a couple of months back, Michael Rosenfeld's lab had published this manuscript where estrogens can also induce double-strand break. So probably, I guess that might happen even in breast cancer. We haven't tested that yet.
Okay. So my third question is probably technical, just a smaller issue. I noticed that if you treat the cells, with the hormone, you activate non-canonical NF-kappa B. But I noticed both P100 and P52 level of both these P100 and P52 are increased. So this indicates that I think this, the hormones increase. It activates, this pathway, but it's increased expression rather than processing of P100.
Yeah, so we have thought about that initially. And you can argue the same thing with STING because we had noted that too. If you compare just the control, band and the P100 and P52 band, the 52 band increases way more compared to the control. So probably there's
one was there's a boss, boss.
Yeah, I totally agree.
Okay, okay.
And Susant, Dr. Kachhap Singh has a question for you in chat.
Sure.
Yeah, Dr. Kachhap Singh, would you like to ask your question?
Yeah, sure. Susant, it's really nice to see you after 20 years.
Yes.
I'll be looking for you. So it's good to catch up here, and I'll follow up with you later on. But I had a couple of questions. One, obviously, the STING pathway leads to C-gas, which is driven by the mitochondrial DNA release. Have you ruled out completely that there is no leakage or the release of the mitochondrial DNA? And the reason I ask because you might have seen our paper that we show that androgen receptor localizes to mitochondria, and we do not know what this function is in the mitochondria itself.
Yeah, so that's a good question. We haven't. So in our search for the cytoplasmic DNA, we did look at mitochondria because we did suspect that selective autophagy of the mitochondria might be happening, and that's another process called as mitophagy. But you're right, we haven't looked very closely to that, and that might be possible too. It might be that even if there are some mitochondria in the autophagosomes, that might also be triggering this process. But we definitely know that gamma H2AX signal is present in the autophagosome, so probably there is some amount of nuclear DNA as well.
It's also, in the context of what you showed about the MAVs, the mitochondrial antiviral, they are localized in the mitochondrial membrane.
Yes.
And that seemed to be playing a very significant role in this pathway, what you showed.
Right.
Yeah, it's really exciting work. I really enjoyed it.
Thank you.
Well, with that's a great note to end this grand rounds on. Thank you very much, Susant, and thank you very much to the department and all the guests who've tuned in for this talk today.
And thank you very much for having me. It's really an honor and a pleasure. And thanks with the slides. Thanks a lot.
Absolutely. Not a problem, Susant.
Thank
Bye.
Bye-bye.